Hey everyone, and welcome. Today, we are going deep, and I mean really deep, inside our own bodies to check out a true marvel of logistics. We're talking about the Golgi apparatus, which is, without a doubt, the cell's master shipping center.
So, let me just kick things off with a question for you. How is it that your body, right this very second, is running a logistics network that makes Amazon's entire global operation look like a lemonade stand? Seriously, it's that much more complex.
And the answer, you might be surprised to hear, isn't some giant organ we can point to. No, it's microscopic. It's happening inside pretty much every single one of the trillions of cells that make you, well, you.
All right, let's get into it. To really get a handle on this incredible system, we've got to meet the manager. And the best way to think about it? Just picture a tiny, super efficient post office. This is the Golgi apparatus.
Okay, so what is it, technically speaking? Well, the Golgi is what we call a membrane-bound organelle. Its main gig is to grab proteins, wrap them up in these little membrane bubbles called vesicles, and get them all set for shipping. It's a huge player in the cell's whole export business, the secretory pathway.
So, what does this little cellular post office actually look like? Come on, let's take a quick tour of the facility and check out the anatomy of the apparatus.
You know, the Golgi isn't just some big round blob. It's actually made of these stacks of flattened membrane pouches, usually about 5 to 8 of them piled up. These folds have a name, cisterna. Think of them as the different departments inside the post office, the sorting rooms, the processing stations, the whole shebang.
And the path a package takes is so incredibly organized, it's beautiful. A protein package arrives from its factory, the endoplasmic reticulum, in what we call the cis-Golgi network. That's the receiving dock. From there, it moves through a series of processing stations, the cis, medial, and trans-Golgi, before it hits the trans-Golgi network, which is the shipping dock, ready to be sent on its way.
So, we've seen the layout, but what's actually going on inside these stations? Well, if the Golgi had a motto, it would be simple: modify, sort, and ship. Let's take a look at the Golgi in action.
This is where the real magic happens, the customization. For proteins, the Golgi is like a master tailor. It can add or snip off little sugar molecules on things called glycoproteins to get them just right. And for lipids, it's more like a specialty manufacturer, adding phosphate groups to build the exact phospholipids the cell needs for its membranes. It's incredibly precise work. But you know, modifying the packages is really only half the battle. Maybe the most mind-blowing job that Golgi has is making sure every single package gets to its correct final destination. This is where it becomes a master of logistics.
So how on earth does it do this with near-perfect accuracy? The solution is just genius in its simplicity. The Golgi slaps a little chemical tag onto each molecule, and these tags work exactly like zip codes, telling the cell precisely where that package needs to go.
And here's a perfect example of one of those zip codes. If the Golgi attaches a tag called mannose-6-phosphate onto a protein, that's it. That's a one-way, non-stop ticket to the lysosome, which is the cell's recycling and waste disposal center. It's a totally clear chemical instruction. No ambiguity.
And this table just lays it all out so perfectly. You've got a lysosomal enzyme, slap a mannose-6-phosphate tag on it, and off it goes to the lysosome. A hormone? It gets a different secretory signal and gets shipped right out of the cell entirely. A phospholipid? It's put on the constitutive pathway and sent to become a new piece of the cell membrane. Different labels, different destinations. It's just an incredibly elegant system.
Now, this whole operation is obviously running on a knife's edge, and it's absolutely vital for our survival. So, what happens when this perfect system fails? Let's talk about a case of total system failure, Wilson's disease.
Yeah, this isn't just some abstract what-if scenario. What happens for real when this finely tuned cellular post office just breaks? Well, the consequences can be absolutely catastrophic.
So, Wilson's disease is a genetic disorder, and what it boils down to is this: the body can't properly get rid of extra copper. This leads to a toxic, dangerous buildup of copper, especially in the liver and the brain.
And the symptoms of this breakdown are just devastating. They can range from these really distinct brownish gold rings of copper in the eyes called Kaiser-Fleischer rings, all the way to complete liver failure, severe psychiatric issues like confusion and psychosis, and just debilitating joint pain.
So where's the weak link? Where's the fault? Get this, it all comes down to one tiny mistake, a mutation in a single gene called ATP7B. This gene holds the blueprint for a critical enzyme whose entire job is to handle copper, and it works right inside the Golgi. When that one enzyme fails, the whole copper export system just collapses.
And because the Golgi can't do its job, it can't load copper onto its main transport protein called ceruloplasmin, doctors can actually see the problem in a blood test. They'll find abnormally low levels of that very protein. That microscopic error inside the cell creates a huge, measurable red flag system-wide.
So let's just put a pin in that. A single clerical error, one faulty enzyme in the cell's post office, means copper can't get the right zip code to be shipped out of the body. And that one tiny mistake escalates into a full-blown systemic crisis.
And that just leaves us with a final thought to chew on. We've just scratched the surface of the unbelievable complexity of one tiny little organelle. So, if the Golgi apparatus is this intricate, this crucial, this beautifully engineered, just stop and imagine for a second what other wonders are hiding inside every single cell in your body right now.